Okay, hi ladies. How are you doing today? I think it's for some of you this may be your first screencast of me. Others it might be your second or third screencast. We'll see how it goes. Okay, so today we are going to go through three or four major ideas, and this should be the last information that's on your test. So we're just going to continue today with a little more on proto-oncogenes and tumor suppressor genes, and those are talked about in 12.4. We're going to do more specifics on mutations from a section 11.6. Talk about tumors, and then finally treatment for cancer. And both of those topics were covered in 9.4. Okay, so we're going to go to page 35 in your packet. So also you should get out some highlighters or some colored pencils or colored markers to do this. So I'm going to flip the screen, but your slide should be page 35 and it should look just like this. Okay, so concept 12.4, you read it a few days, probably maybe even a week or more ago at this point. Its genetic changes contribute to cancer. So we know cancer is uncontrolled cell division at this point, and we know that it results from mutations, and it's mutations in genes that regulate the cell cycle. So let's just take a look at this figure here. We have 4 different chromosomes. So a good little point of review here is that none of these are homologous chromosomes, meaning there are no pairs here. There are 4 different shoes or 4 different chromosomes. And so we see 4 different chromosomes here, and there are 0 mutations, 4 normal chromosomes. And so what happens to cells that have those normal chromosomes is that they're completely normal. And these cells just happen to be found in the colon wall. So that's the large intestine. And so there's no cancer here, no mutations in genes that control the cell cycle. So they have perfectly normal cells. Also this information was shown to you in a different way in those cell cycle animations from the NIH that we did a couple days ago. So animation 3 and animation 5 have the same information. We're just showing it in a different way, the same way the way it's shown in your textbook. And then we also went over this table, which showed more specifics about tumor suppressor genes and proto-oncogenes. So just to remind you that the information that you saw in these 3 images is shown just in a different way down here. Okay, so getting back to our normal cells and our normal chromosomes. Now let's look what happens when we have a mutation. So here, this little yellow band represents the conversion of a normal gene to an oncogene. So this would be a proto-oncogene that has now turned into an oncogene. So you can go ahead and highlight that little piece yellow and then maybe highlight the word oncogene. So there's only 1 mutation here, and you can see that there is some increased cell division. So these cells shouldn't be dividing at this point, but they are. And you can tell they're dividing because of the way the DNA looks. So here in this cell right here, the DNA is in chromatin form. And here in this cell happens to be in mitosis, and its chromosomes are lined up in the middle. So when you look at cells under a microscope, you can tell that they're cancerous or precancerous because if there's a large number of them that are in mitosis. Okay, so here we have increased cell division. Now let's see what happens when we have 2 mutations. So we're kind of going in chronology here in a timeline. So this is like time 1, this would be time 2, and then here would be time 3. So if you want to draw an arrow that says later, even later, to show that there's really an accumulation of mutations over time. So you could even put a big arrow going across the screen. Oh, let's see if I can use my stylus that I just got. Do you think it's possible? You think it's going to work? I don't think I have it plugged in. Okay, that might be some homework for Mrs. Doherty. We'll try that. Okay, so anyhow, time is on the x-axis. So this is later, and this is earlier in history. So here now we have two mutations. So here's a mutation, that previous mutation that existed in the and became an oncogene, that proto-oncogene became an oncogene, and then up here now we have an additional mutation. So that is a tumor suppressor gene that got mutated. So go ahead and highlight that yellow as well. And you can see now that we have two mutations, there is an abnormal ball of cells growing here. So maybe this could be a precancerous cell, and there's a tumor right here starting to form. And then even later in time, we now have 4 mutations. So go ahead and highlight this chromosome has another mutation. So now we see 4 total mutations. And now this is truly a malignant tumor. And we'll get into malignant in a couple slides. So you can see this is where the cancer started, and now it's invading other tissues. So this is probably going to spread to another area of the body. So again, just showing that rarely is cancer a result of one mutation, but normally it's the accumulation of mutations that really lead to cancer. Okay, let's go on to our next slide here. Okay, so 11.6. This is page, it's actually page 37 in your packet. It actually doesn't have a number, so it's the very last page in your packet so far. Okay, so I don't know if you can see this up here. I already have the screencaster set. Concept 11.6, it's mutations can change the meaning of genes. Okay, so you're going to stay on page 37 here, but I'm going to advance to the next slide just to zoom in on what we see down here in the bottom. Okay, so what we're looking at here is the DNA sequence for a normal gene. So here are the DNA nucleotides. So this is a piece of DNA. And we know from section 11.4 that in order for DNA to be expressed, meaning for a protein to actually be made from that recipe in the DNA, it requires 2 steps. So the first step is translation, which isn't, or excuse me, transcription. Transcription is the first step, and that's not shown here, but mRNA, the message of DNA, messenger RNA, is made via transcription. So here's the corresponding messenger RNA that would be made from this sequence. Okay, and then the second step would be here, translation, and here is the protein or the polypeptide chain. So here are the individual amino acids that are coded by each codon here. So this codon AUG codes for methionine. And then these next 3 nucleotides, AAG codes for lysine, so on and so forth. And right here, we're looking at 2 different kinds of mutations. So take your marker, doesn't matter what color, or a highlighter, and I want you to highlight this T here, and this A, and then this amino acid in this section of this figure here on the left. So you can see right here in the original DNA sequence, there should be an adenine, but here we have a mutation, and the mutation is just a change. Any mutation is just a change in DNA, and this is what we call it's a very simple kind of mutation, and it's just called a substitution. So let's go ahead and write substitution down there. So you should have an adenine in this DNA sequence, and you don't. There's a thymine. Well, that's going to change the mRNA sequence, which is going to alter the amino acid. So instead of having a glycine, there's a serine in the polypeptide chain. And so a substitution sometimes can result in a modified protein. So the protein structure is taken shape. So we went over this example last week or so, where we looked at regular hemoglobin and then sickle cell hemoglobin. And you can see in hemoglobin there should be a glucine, and now there's a valine. So because of a substitution, it changes the amino acid sequence, meaning it changes a specific amino acid in the protein or in the polypeptide chain here. And now you have a genetic disease just because of one single nucleotide mistake in the DNA sequence. So again, this would kind of be like if your recipe for a cake said to bake at 375 and you baked it at 475. There's only one number difference in that recipe, but clearly your cake is not going to come out okay. Okay, and now the second type of mutation that we see over here is what we call a deletion. Okay, so you can see in the original amino acid sequence, where are we? Lysine. There should be a CAAA. And if we go down here to this nucleotide sequence, it's CAA. And then look, a nucleotide, an adenine, was deleted. So we call that kind of mutation a deletion. And that's going to completely alter the polypeptide or the protein that's formed. So instead of having a phenylalanine, a glycine, and an alanine, you have a leucine, an alanine, and a histidine. So this is going to completely change the protein. And I know over here with sickle cell anemia, we see an example of a mutation that is harmful, and you don't want this kind of mutation. And I just want to make you understand that not every mutation is a bad thing. Mutations could be positive, they could be negative, or they could have no effect. Now this deletion right here, some deletions could result in a complete change in an organism's body type. So this is just an example of a fish that lives in the Pacific Northwest. And you can see down here it has this little fin. It's called a pelvic spine, actually. Here's the fin. This is called a pelvic spine. So this is how a fish looks, the original historical or original stickleback. And then a deletion mutation actually caused that spine, that pelvic spine, not to be formed here. And this actually benefited the organism because then it couldn't be grabbed by predators. So in this case, an organism had a mutation, a deletion, and it actually was helpful based on the environment that it lives in. So mutations aren't always detrimental to the organism. They can be positive, they can be negative, or they could have no effect on the organism. So that's just two examples of mutations, substitutions, where the wrong nucleotide is put in when DNA is being replicated, or perhaps a nucleotide is left out, and we call that a deletion. And again, you should highlight in whatever, if you want to do a different color to show you that deletions are not the same thing as substitutions, you can go ahead and do that. Okay, and then there aren't always, the third kind of mutation I want to go over here has to do with ultraviolet radiation. So here we have DNA, and you can see the double helix, and now we see this incoming ultraviolet radiation taking place. And what actually happens when you expose your skin to ultraviolet radiation is that this UV radiation can actually cause a little kind of bubble over here in the double helix. And so what does that look like on a molecular level? Well, it kind of interferes with the base pairing. So this C should be base pairing with G. This A should have a hydrogen bond, or this T and A should have a hydrogen bond. And then this T and A should have a hydrogen bond. And then again, this G should have a hydrogen bond over there. But this ultraviolet radiation causes the T's to have a bond. And it causes this bubble. And you can imagine that as, DNA polymerase is chugging down this strip of DNA, when it comes to this bubble, it's like confused. It's like, what do I do? What do I do? Because this is clearly misshapen. So this isn't a substitution, it's not a deletion, it's merely a change in DNA causing that bubble. And remember, ultraviolet light is something that's really high energy over here, so that's causing, you need a lot of energy to cause those two thymines to form that bond. And that's 3 kinds of mutations. Okay, and our next video is going to talk about where and when mutations happen matters.